So thank you very much for that introduction. I'm going to give you just a brief introduction to my work of the gut microbiota, fecal short-chain fatty acids, and cardiometabolic risk in these 5 African populations that we've been studying for over 10 years. So I just would like to also thank my colleagues from Ghana, South Africa, Jamaica, Seychelles, and the US, and I will introduce them. So the modeling the epidemiologic transition study actually started over 10 years ago in Ghana, South Africa, Jamaica, and Seychelles, and the US. And in Ghana, we work out of Nkwantakesi, which is a rural village about 25 kilometers outside of Kumasi. And we're in South Africa, we're based in Khayelitsha, which is a peri-urban informal shanty town, part of Cape Town. Our work is based out of Kingston in Jamaica, and on the main island in Seychelles, Victoria. And then finally, our US site is in a town on the western suburbs of Chicago. And what we have used is the World Bank's Human Development Index to classify each of the countries as they span the epidemiologic transition. Professor Amy Luke started the study over 10 years ago because she was interested in understanding factors that contribute to cardiometabolic risk and diabetes in these 5 African origin populations. So just to let you know, over 10 years ago when we started this study, the prevalence of overweight and obesity was very low in Ghana, in both men and women, and highest in our US participants in both men and women. The South African women have also presented with a very high prevalence of obesity back in 2010. So we discovered that over 10 years ago that obesity drives disease. And so if you look in Ghana, where they had very low prevalence of obesity, their diabetes and hypertension was very low. And then if you go all the way to the right side of the screen, where obesity in our US cohort was over 50% already 10 years ago, to diabetes and hypertension is exceptionally high. As part of the measurements in our studies, we did 2 structured 24-hour recalls, and that allowed us to capture very accurately the dietary fiber intake in the diets. And what you see is that people who live in Ghana are consuming almost twice as much dietary fiber than people who particularly live in the US. And most striking is the proportion of both the soluble and insoluble fiber intake. When you actually look at where the foods are coming from, in Ghana, obviously the more traditional diets with light soup, banku, yams, compared to in the US, the top foods that we found were actually candy, chicken, white bread, French fries, and cookies. And that was really contributing to the differences in the fiber intake. So what we found is that dietary fiber is very well correlated with cardiometabolic risk. And so this looks very busy, but I just want to draw your attention to if the bar is orange, that means that that group is the lowest quartile of fiber intake. If the bar is green, that means that's the highest quartile of fiber intake. And all the way on the right, when we pool all the sites, we see that those people who are in the lowest quartile of fiber intake have significantly more metabolic syndrome, high CRP, and CRP, remember, is an inflammatory marker, and there are many more people that are obese in the low fiber intake group than in the high fiber intake group. So I was really interested in this work. We published this back in 2018 already because the gut microbiota are important for digesting the fiber intake. So my colleagues, Dr. Brian Layden and Dr. Jack Gilbert, we wanted to see, well, who is already doing international studies looking at the gut microbiome and how it can relate to obesity in international populations. And way back in 2016, there were only 5 studies that were really looking at differences in the gut microbiome across different populations. So Yatsenko was doing Kenya and North America. De Filipino was doing North America and China. And Carson was doing the Asia with Italy, with the Italian populations. And so we knew with our 5-country study, looking specifically at African origin populations, we would be able to really understand the role of the gut microbiota with fiber intake. And so we developed the METS microbiome study, which was specifically to look at gut microbiota, the fecal short-chain fatty acids, and obesity risk in these 5 populations. And so our aim was to define the associations between the gut microbiota, short-chain fatty acids, and obesity in these 5 populations spanning the epidemiologic transition. And we used the same 5 sites. And originally, we had recruited 500 people over 10 years ago in each site for 2,500 people. When we went back in 2018, we recruited 400 in each site. So our sample was over 2,000 people that we were collecting data on. And it's very clear. So I want you to start on the left side of the screen. The green is the participants from Ghana, orange is South Africa, purple is from Jamaica, Seychelles is pink, and US is green. And this is in the middle bar, this is alpha diversity. And alpha diversity describes how many different species of bacteria there are in your gut. And so the higher that number, the better it is. It means you have a very diverse gut microbiome. And so you can see that the participants in Ghana have the highest diversity in their gut microbiome compared to the participants in the US who have the lowest diversity in their gut microbiome. And then when we look at the gut microbiome by whether someone is obese or not obese, we find, if you look over here, there are significant differences between people who are obese versus not obese, with people who are not obese having a more diverse gut microbiome. And this supports previous research that shows that people who are not obese have a very diverse gut microbiome. It's very robust. So, and normally we look at 2 measures of the gut microbiome. We look at alpha diversity, which is how many species there are in the gut. And then beta diversity tells us more about the population of the bacteria and how they cluster together. And what we actually see is when we pool all the 5 sites, this was very exciting. There are 23 unique species that are associated with obesity in the gut microbiome. And this allows us really to target functional work to understand what is the role of those species in metabolic pathways that may be driving obesity risk. Not just obesity, though, we are also able to look at the species and how they are associated with type 2 diabetes. And on the right here, we find that the species of bacteria in your gut are able to predict whether you have type 2 diabetes with about 63%. And that's really quite good. And about 65%, the kinds of species you have in your gut can predict whether you have obesity with about 65%. So we really know that the types and kinds of bacteria that are in your gut are really associated with whether you have obesity or type 2 diabetes. We were also interested, obviously, in the short-chain fatty acids. Short-chain fatty acids are critical for life. They are receptors all over our body, from the pancreas, the liver, bone, white adipose tissue, brown adipose tissue, and they're vagal nerve and in the brain. And they are associated with thermogenesis, how we sense our food, our feelings of fullness. And what we find is that obviously those participants that are consuming the highest amount of fiber in their diet have the highest levels of these short-chain fatty acids in their stool. And those participants from the US that have very little fiber in their diet, they have the lowest short-chain fatty acids present. And obviously, those short-chain fatty acids are associated with gut diversity. So on the x-axis over here, this is the diversity of the gut, so higher is better. And then on the bottom is the short-chain fatty acid levels. And you can see that it is a positive relationship, meaning that the more short-chain fatty acids you have, the more the diversity of the gut microbiome. And we particularly want to look at butyrate. Butyrate has been identified as having significant health benefits, all linked to feelings of fullness, satiety, thermogenesis, and body fatness. So I want to acknowledge my collaborators from Loyola University, from Kwame Nkrumah University of Science and Technology, the University of Cape Town, the South African MRC, the University of West Indies in Jamaica, Ministry of Health in Seychelles, University of Illinois. I worked very closely with Brian Layden, who's critical for the short-chain fatty acid work, as well as Jack Gilbert at the University of California in San Diego. I'm funded through the National Institutes of Health, the AXA Research Fund, as well as the Wellcome Trust, and I have no conflicts of interest to declare, and I'm happy to take some questions.
Yeah, let's give a hearty clap.
Thank you, Prof, for your presentation. My question is, you said the fecal short-chain fatty acids were determined, was it from the fecal, or how did you collect that data? And also, were you taking into account that whether the short-chain fatty acids were being absorbed and the absorption, if really you are taking it from the fecal? And then the other question is also, how were you determining the gut microbiome? Yeah.
Okay, those are all great questions, and I'm sorry, I should have started off with that. So for all of our studies, the participants provide a stool sample, and from the stool sample, we then we send a sample off for genetic sequencing to understand what the species are in the gut. Okay, so you send a sample off to find out what genes are in that stool sample, and those tell us about the kinds of bacteria that are in your gut. And then we also send a sample of the stool sample for metabolomics. And the metabolomics tells us about the amount of short-chain fatty acids that are in the stool sample. So that is how we get the sample, and that is how we do the measurement. Then you are absolutely correct that we measured in my study the fecal short-chain fatty acids, and we use that as a proxy to infer what the blood levels were for the short-chain fatty acids. And so you can do two things. You can either do the blood levels or you can do the stool levels, and we're assuming that the two are quite closely correlated. And so whether the amount of short-chain fatty acids that you see in the stool sample is directly proportional to the amount of short-chain fatty acids that you have in your blood, that is a great question. And actually what we are studying now to see whether the short-chain fatty acids that you have in your blood is representative of the same as in your stool sample. But certainly we know that the people who eat more fiber in their diet definitely have more short-chain fatty acids in their blood and in their stool samples.